Okay, my name is Reena, and this is our second lecture regarding the phylum Porifera. And in this lecture, we are going to discuss about the canal system in sponges. So basically, there are three types of canal system present in sponges. From simpler to complex, they go like Eskinoid canal system. Second one is Cycanoid canal system. Third one is Leukophilic canal system. This goes from simpler to complex. Okay, why do we study canal system is because everything that a sponge does, all the function, they are on a cellular level. Okay, whether be it your nutrition, excretion, reproduction, they will depend upon the water, like whatever the surrounding water brings into their body, they are going to absorb all of those materials which they require for their survival, and the rest of the excreta and the waste material, they will expel it out from the osculum. So this is the simplest canal system, which is Eskinoid canal system, and this is found in Leukosolania. These are your porocytes. This one is your ostia, dermal ostia. Okay, and from here water is entering. The water available in the surrounding is entering through this ostia and coming into the spongiocele, and in the spongiocele this choanocyte will absorb all the nutrition from this water, and all whatever, if they are doing their reproduction and all, they will absorb the gametes that are coming in with this water, and then they are going to secrete the excreta or waste material into this water, and this water is going to come out of this osculum. Okay, and this outer wall is pinacoderm, and pinacoderm is lined with pinacocytes, and this one is mesoglia, in which you have spicules and this amoeboid cells. Okay, and then the inner wall is choanoderm, which is lined by choanocytes. Okay, this one is the simpler canal system. Just to make the pores more visible, this is the basic diagram of Eskinoid canal system. Now the water from the surrounding medium will enter first through this dermal ostia, and then this water is going to get absorbed by these choanocytes, and then the waste material will be dissolved in this water, and that waste water is going to be expelled out from this osculum. So this is the path of water current in the Eskinoid type of canal system, and example is Leukosolania. Second type of canal system is Cycanoid canal system, and it is found in cycone. This is the cycone body, okay, and we are taking this much part of this cycone and looking it in an enlarged view. In the Eskinoid type of canal system, you had this body wall, a simpler body wall, right? In case of Cycanoid, this was your Eskinoid canal system, but in case of Cycanoid canal system, this body wall is going to get folded, like invagination and evagination will occur here. Okay, basically this invagination and evagination of choanoderm, this why this happens, to increase the surface area for the absorption of water. And in this folded choanoderm, you are going to see some slits, some pores opening in this radial canal only. Choanoderm, which is going to have some pores. These are the contractile cells that are present in between. These are called myocytes. So this part here, this opening, this is your dermal ostia, and these dermal ostia leads you into the incurrent canals. So here also there will be dermal ostia. Dermal ostia is the passage that is present in between these myocytes from where water can come in. Okay, this dermal ostia will lead you into the incurrent canal. So this invaginated fold of pinacoderm, this makes up the incurrent canal. After that, there are pores that are leading you into the invaginated choanoderm. Okay, now this one inside it is invaginated choanoderm. These are called radial canal. So water is entering into these radial canal through another opening which is called porosopyle. Okay, so poroso means near and pyle means gate. Okay, gate means the wall, body wall. Okay, the pores which are near to the body wall are called porosopyle, and the ones which will be away from the body wall, they are going to be called apopyle. Okay, so from the porosopyle, the water is entering. The water is entering into the radial canal. Okay, so this one here you have porosopyle. So inside the radial canal, you have choanocytes, and they are going to absorb the water because the number of choanocytes here have increased due to the enfolding of this choanoderm. So more absorption of water will be there, and after that water have absorbed the nutrition and all and excreted all the waste material into the water is going to go in apopyle. So this one right here, this opening is apopyle, which is away from the gate or body wall. Okay, apo means away, pile means gate. So this apopyle, how it looks like is, this is your apopyle. This apopyle has This apopyle is surrounded by four myocytes. These myocytes are basically contractile cells which actually determine the diameter of the opening of this pore, and in between, and this one is diaphragm. Surrounding to this myocyte is diaphragm, which is basically provided with some fibers. So these myocytes along with these fibers decide the diameter of this apopyle opening, or this apopyle is going to push water into the spongiocele. Okay, the spongiocele, from spongiocele the water will move out of this sponge body. Okay, from spongiocele out through the osculum. Osculum is also going to have these myocytes which are going to determine its diameter. So this was your cycanoid type of canal system which you see in Cycone. Now you have the most complex type of canal system which is Leukophilic type of canal system, and it is found in Euspongia or Spongula. The freshwater sponge which you study as an exception, it comes under this canal system, Leukophilic type of canal system. What happens is the infolding and outfolding of that choanoderm that you saw in case of cycanoid canal system. This is your cycanoid type of canal system, and before this we studied Eskinoid type of canal system which was simple lining of choanocyte, and now this Leukophilic type of canal system, what happens is there are further branches in this radial canal. Okay, what happens is, assume this radial canal, it is going to get branched like this. Let's look at it. It is getting branched, further branched. Why it is getting branched? To increase the surface area. for the absorption of water. Okay, this is your first radial canal, and the second radial canal would be folded in the same manner. They could be attached to each other or they could be simply folded. Sorry, this is the waterfront here. It is going to enter through the dermal pores. Okay, this is your ostium, or you can also call it dermal pores, dermal pore. Okay, from the dermal pore, it is going to enter into the incurrent canal. These chambers, they are actually surrounded by these myocytes. Okay, this is your sponge body wall. So this is your ostium or dermal pore from where water enter, and this one right here is your incurrent canal. What is incurrent canal? It is the infolded, invaginated pinacoderm. Okay, this one is your pinacoderm, so it has been invaginated to meet this incurrent canal. And from the incurrent canal, you are going to see these porosopyle. You can also remember this as poroso means pre or prior. Okay, the pores which come first, the pores which encounter with the water current first are porosopyle. So from the incurrent canal, the water is going to enter into this radial canal. This is your radial canal through porosopyle. And in the radial canal, we are going to see choanocytes. Okay, and these choanocytes are going to absorb the nutrition from the water current and excrete the waste material, same as in cycanoid type of canal system. And then that waste is going to come into this excurrent. This is your excurrent space, okay, or excurrent canal. This is going to come into excurrent canal through this one. This opening is apopyle, and then this water current is going to move out of this sponge. Here we are going to see a man canal, which is going to combine all these branched radial canal, and this water current is going to move out of this man canal and finally through the osculum out of the sponge body. Okay, so this was your leuconide type of canal system. The leuconide type of canal system is further divided into aphodal and diplodal type of canal system. In case of aphodal, what we see is the apopyle. The apopyle is longer in length. And in case of diplodal, what happens is, so this is your first branched radial canal system. First branched radial canal system is attached. So the first branched radial canal system is attached to the second branched radial canal system with help of, this is your prosopyle. Okay, and in case of the diplodal type of canal system, both so this type of canal system you see in spongilla as well, the freshwater sponge. Okay, the sponge which is toxic or harmful, that is Cleona. It destroys mollusks and also destroys rocks.